stomach, or this is esophagus, excuse me. Stratified squamous, non-keratinized epithelium. Cells are alive all the way out to the surface. We come over this way as the stomach-esophagus junction, and as I mentioned, we see this is the opening of the gastric pit. That's a gastric pit. These are mucous neck cells. The nucleus is always to the base. And just mucin droplets up here. The nuclei sit down at the base. Now we look at that, and it's cut 90-degree different across the way. And we'll be looking down into it, and that's what we see right over here. Same thing. So nuclei to the base, and you can see how closely arranged these are. So those are the gastric pits. If you could get small enough to walk in on the surface of the stomach, that's what it would look like. You'd just be looking down into these openings. They go some distance down into the tunica mucosa, like these do, and then they branch. So they're out of the plane section. They branch into the gastric glands. The gastric glands, we see on the next slide. So we'll only the only place where we'll look for all four tunics is in the jejunum of the small intestine. So this is the tunica mucosa. These are the gastric pits. This is the gastric gland. These are the gastric glands. So you really get the flavor for, with the whole tunica mucosa from, that's the lumen of the stomach up there, from here all the way down here, and now we're going to get into connective tissue. So that's all tunica mucosa. Here's the gastric glands here. The gastric pits only go a little ways in. So we see two different categories of cells here. The ones that are fairly plain looking in the cytoplasm, large spherical nucleus, kind of in the center of the cell, those are parietal cells. So they're responsible for the production of hydrochloric acid and intrinsic factor. And the darker, chunkier looking ones, those are the chief cells, and they're going to be making the inactive protease pepsinogen. And they stain dark because they've got a lot of ribosomes in order to make that protein. Okay, so once you see villi, and I've got a handout for you today that I'll give you in class, so you can kind of ask yourself, where am I in the digestive tract? When I see this, the first is that. And so if I see the first thing, then I ask myself a couple of other questions. So you kind of just go through that as you're trying to assess what happens to be here. So we see villi, we know we're in small intestine, and our next question is, where are we? So let's think about what the villi look like. If they're spiky, we're in the duodenum. They're not very well developed. And then we would look at the second tunic, because this is the tunica mucosa. We look at the second tunic, the submucosa, and ask if there are Brunner's glands there. And they're everywhere, so you don't have to look around very hard for them. If you're in the jejunum, which is what this is, then we have kind of the rounded ends to the villi, simple columnar epithelium, lamina propria, the villi end here, but the mucosa goes all the way to this point, and we've got crypts of Leber present. If we're in, and if we don't have anything special in the submucosa, we'll look around, we'll see that. If we're in the ileum, the villi are stubby, block-shaped, and on one side in the submucosa, we've got Peyer's patches, unencapsulated lymph nodules there. So there's the villus right here, kind of finger-like. If we look at this kind of view, we see it's a simple columnar epithelium, and there are some goblet cells present here. If we were to compare slides looking all the way down the tube, the further we go down the tube, the more goblet cells we have. Lamina propria is to the center. We're not going to try to identify anything specific there. The villus ends here, but then we've got these intestinal crypts. We backed off a little bit here so we can see that. So the intestinal crypts are still going to be a simple columnar epithelium. It just increases surface area for absorption. And the intestinal crypts are between every villus. And then we come over to the submucosa. It's dense irregular connective tissue. Blood vessels abound. We also have, some fairly large cell bodies of autonomic nervous system neurons, but they're not on your list, so we're not going to worry about it. Dense irregular connective tissue and a lot of fibroblasts. The third portion of the tunica mucosa is going to be the muscularis mucosa. And here we see the smooth muscle cells of the muscularis mucosa. That means we're out of the mucosa, now we're in submucosa, which we just did here. And then we'll continue out and we go into the muscularis, inner circular, outer longitudinal. And the profiles we see here are the nuclei of the smooth muscle cells. And here the muscle cells are going in and out of the plane of the board. And we see the dark profiles of those muscle cells. And the outer layer is the serosa. And there we see that serous membrane. Now, if we're in, that was jejunum. Oh, I didn't go all the way around. Let me do that. So let's watch. Here's the submucosa, and let's watch as we go around. I mean, I can go to higher power, and you can see there's no glands there, and the Brunner's glands that we see in the duodenum are everywhere. You don't have to look very far for them. So there are no glands present here, and clearly no Peyer's patches. So I've gone down one side, coming up the other. So that says I'm in jejunum. If I look for duodenum, okay, the spiky nature to the villi is an immediate indicator that I'm not doing much absorption here, and we don't do much absorption in the duodenum because we haven't finished digesting everything down to monomers. So if I think again, the assessment is if I see villi, then I think small intestine, now which part? So my attention is immediately thrust to the submucosa. And here we are in submucosa, and there's Brunner's glands. And they are everywhere, all the way around. I haven't tried to show you each space, but guaranteed, they are everywhere, all the way around. So that's simple cuboidal epithelium of the gland, and we actually see some of the duct going on its way out, like right there. So that tells me I'm in duodenum. Now, if I think I see kind of stubby looking villi, which is what these are, then I think maybe I'm in ileum, and I look at the submucosa. So here's the submucosa, and I could go to lower power or higher power and demonstrate to you that there's no glands present there. There aren't any. But now I come up around like this, and there's Peyer's patches. 1, 2, 3, 4. Unencapsulated lymph nodules, and they're always on the side where the serous membrane is attached. So it's pretty easy to differentiate between the different regions there. The only place where I'm going to ask you all four tunics is when we're in the jejunum, because we see everything best there.